is talk about a model for neuropsychiatric lupus. And I'm going to present some mouse studies, and I'm going to present some human studies. And my goal is to convince you that, in fact, it is possible to study neuropsychiatric lupus, especially to study the cognitive disorder in neuropsychiatric lupus. And this is something that must be done. So I have a slide of disclosures. I consult for many companies. None of these consultancies are related to the work that I'm going to talk about. So we know that lupus is a disease of protein manifestations, and that wherever we know something about the tissue injury or the cellular injury in lupus, what we know is that it begins with autoantibodies. And anti-DNA antibodies are the most common anti-nuclear antibody in lupus, and they are diagnostic of the disease, and they are also pathogenic in the disease. So neuropsychiatric disorders are among the disease manifestations, and these were really long neglected. But in 1999, there was a consensus conference where a group of individuals, and I was not among them, identified 19 different symptom complexes that could be attributed to lupus if there was not, in fact, another etiology present. And what you see in this slide is that when cohorts of lupus patients at different places in the world have been examined, the incidence of neuropsychiatric lupus is quite high. It goes from about 50% to over 90%. The Halifax cohort, where the incidence is lowest, is because they actually don't include cognitive disorders, cognitive impairment among the symptoms of neuropsychiatric lupus. What you also see is that cognitive dysfunction is the most common disease manifestation. And a few years ago, the FDA conducted a focus group with lupus patients, and they identified cognitive impairment as one of the 3 symptoms of lupus that most detract from quality of life. So cognitive dysfunction is common, and it's common even in inception cohorts of lupus patients. And it is a manifestation that matters to patients. So we got involved in thinking about neuropsychiatric lupus and cognitive dysfunction in lupus when we were looking to see if a mouse monoclonal anti-DNA antibody might actually be able to bind a protein antigen. And what we found were peptides that were bound by this anti-DNA antibody. And we identified the consensus peptide that you see above. And when we looked to see what proteins in the body had this consensus peptide within them, we were excited to find that, in fact, the GLUN2A and GLUN2B subunits of the NMDA receptor both have this consensus sequence within them. So what are NMDA receptors? Well, they're receptors on neurons that are made up of 2 obligatory GLUN1 subunits and 2 of any 4 GLUN2 subunits. Glutamate is the ligand for the NMDA receptor. And when glutamate binds, a channel in the receptor opens to allow the influx of calcium into the cell. And this process is critical in learning and memory, but excessive engagement of the NMDA receptor leads to too much calcium entering the cell, and that causes cell death that is called either excitotoxic or glutamatergic cell death. So we wanted to know if antibodies with this cross-reactivity that bind DNA and the NMDA receptor, and that we call DNR antibodies, are in fact present in lupus patients. And what you see on the left are serum titers of these antibodies, showing that they are indeed present in lupus patients with neuropsychiatric manifestations of lupus. And on the right, you see that there are high titers of these antibodies in the cerebral spinal fluid of patients who have non-focal central nervous system manifestation of lupus, above what you see in the cerebral spinal fluid of lupus patients with no neuropsychiatric lupus or with peripheral nerve manifestations of lupus. So these data said to us that this is an important antibody specificity. I should say right away that these are not the anti-NMDA receptor antibodies that are present in individuals with limbic encephalitis and were made famous in the book Brain on Fire. Those antibodies bind to the gluN1 subunit, and when they bind, they cause receptor internalization. The symptoms are usually transient because once the antibodies are no longer present in the brain, the receptor is re-expressed at normal levels on the surface of neurons, and there's normal neuronal function. And you will see that's not what we see with these anti-gluN2A and gluN2B antibodies that are present in lupus patients. So we wanted to develop a model where we could study these antibodies. But if you look at mice with spontaneous lupus-like disease, they have many anti-brain antibodies and they have many cytokine perturbations. So we wanted to develop a mouse that, as its only immunologic autoimmune manifestation, had these antibodies present at high titer in serum. So we immunized non-spontaneously autoimmune mice. And what you can see is if you immunize with the peptide on a polylysine backbone, which we call MAP peptide, the mice develop high titers of anti-DNA antibodies. If you immunize with only the poly-L-lysine backbone, they do not. The titers of the antibodies are equivalent to titers in one of the most studied mouse models of lupus, the NZBWF1 model, and these antibodies deposit in glomeruli, so they have the critical hallmark of a pathogenic anti-DNA antibody. But when we looked in the brains of these mice, they were completely normal. And we reasoned that this is, in fact, because the mice had an intact blood-brain barrier. But it is clear that there are many insults that can alter blood-brain barrier integrity and allow antibodies to penetrate the brain. And in fact, there are more that are discovered all the time. Inflammatory mediators that may be present in patients with infection or with a lupus flare can impair blood-brain barrier integrity, as can hypertension, smoking, and other toxins, and can stress with its high levels of epinephrine. And these insults actually alter the blood-brain barrier in a region-specific manner. So we developed a model where we immunize either with the peptide on a polylysine backbone or the polylysine backbone alone to either develop the lupus antibody or not. And then we give the mice lipopolysaccharide, which causes a breach in blood-brain barrier integrity in the hippocampus, so that antibody penetrates into the hippocampus. And that antibody either includes this lupus specificity or does not. And what you see in those mice are two stages of brain pathology. An acute stage that's characterized by neuronal loss, and a chronic stage that we don't see beginning until at least 4 weeks after LPS, that's characterized by microglial activation and loss of dendritic arborization of hippocampal neurons. And what you see in the insert on the lower right is that by 2 weeks after LPS, there is no more antibody detectable in the brain. So this second phase of pathology arises after the insult is no longer present. And this just shows you the loss of hippocampal neurons that happens acutely. And this shows you that at 2 weeks of age, 2 weeks after LPS administration, there is no loss of dendritic arborization in the DNRAb positive mice, but at 4 weeks and then 8 weeks after LPS administration, there is a loss of dendritic arborization. And this has functional consequences. So hippocampal neurons are responsible for generating spatial maps. They have what's called place field activity. And what this means is, as you navigate an environment, a particular hippocampal neuron will fire at every location that you move into in that environment. And the size of the location over which the hippocampal neuron will fire is called the place field size. And what you see is in mice that were immunized, but not with the peptide, just with polylysine backbone, and were given LPS, and in mice that they have exactly the same place field size of their hippocampal neurons before and after antibody penetrated the brain, before and after LPS administration. In mice that had lupus antibody penetrate the brain after LPS was administered, you see an enlarged place field size. So the particular hippocampal neuron is firing over a much larger area in the spatial arena. And that means that the spatial map is of lower resolution. So we looked to see if this was actually the case. And we subjected mice that either had normal immunoglobulin penetrate their brain or immunoglobulin, including the lupus antibody, penetrate their brain, to an object memory test and a spatial memory test. So in the object memory test, you put a mouse in an arena with 2 objects. You take the mouse out, and then you return the mouse after you've changed one of the objects. And the mouse that had normal immunoglobulin penetrate the brain and the mouse that had lupus antibody penetrate the brain both spend more time exploring the novel object than the familiar object. So they have normal object memory. You can also ask about spatial memory in mice. When you put a mouse in an arena with two objects, you take the mouse out, you move the location of one of the objects, and you put the mouse back in. And the mouse that has no lupus antibody penetrate the brain, just normal immunoglobulin penetrate the brain, will spend more time exploring the object that's in a new location. Whereas the mouse that had lupus antibody penetrate the brain will not. So the mice with lupus antibody penetrating the hippocampus have a selective defect in spatial memory. If you look at these mice, you can also see they have increased microglia. So this is the mouse that had just normal immunoglobulin penetrate the brain, and below it is the mouse that had the lupus antibody penetrate the brain. And in red, you see microglial cells, and you can see that there is a microgliosis where the lupus antibody penetrated the brain. Now, microglial cells, when they're quiescent, are lacy-looking cells. And when they are activated, become compact with stubby appendages. And you see that in the mouse that had normal immunoglobulin penetrate the brain, the microglial cells at 2 weeks after LPS and 8 weeks after LPS have a lacy phenotype. Whereas the mice that had the lupus antibody penetrate the brain have this stumpy, activated morphology, and that can be quantitated as you see on the right. And this is a PET scan with a ligand, using a PET ligand that binds activated glial cells, and you see increased binding of the PET ligand in the mice that had lupus antibody penetrate their brain compared to the mice that had normal immunoglobulin penetrate the brain. And we were interested if these activated microglia might in fact be responsible for the loss of dendritic arborization, because microglia can engage in an activity called pruning, where they actually eat away at the synapses, causing the dendrites to retract. And so what we did was treat these mice with a CSF1 receptor inhibitor that was present in their chow. And one of these inhibitors, 5622, crosses the blood-brain barrier. And so the microglial cells are exposed to the inhibitor. And the other inhibitor, 73086, does not cross the blood-brain barrier, so does not affect microglial cells. And you see on the right, on the far left, that there was substantial depletion of microglial cells fed with the inhibitor that crossed the blood-brain barrier, and much less depletion with mice fed the inhibitor that does not cross the blood-brain barrier. And if you allow the microglia to reconstitute, you stop feeding the CSF1 inhibitor, feed normal chow, and allow the microglia to reconstitute, you can see that where there was depletion of microglia, they now reconstitute with microglia and have normal neuronal dendritic arborization, whereas those mice that didn't deplete and therefore did not reconstitute, have the same microglia present, still have a loss of dendritic arborization of the neurons. So the microglia are essential for the loss of dendritic arborization, because they are engaging in this process of pruning. So how does pruning work? Well, pruning works because the microglia are targeted to the neurons by C1q. And what you see here is much more C1q present in the brains of the mice that had the lupus antibody penetrate, and that is true both presynaptically and postsynaptically. And what you see here is that in the mice that had lupus antibody penetrate the brain, there's actually increased production of C1q by microglia. So the microglia are in red, the C1q by in situ hybridization is in green, and yellow is where there is C1q being made in microglia. And you can see that there's much more when the lupus antibody penetrated the brain. And if one generates this model in mice that lack C1q, what you see on the left is that the first stage of brain pathology with neuronal death is in fact present, but there is no second stage of neuronal pathology. There's no loss of dendritic arborization, and the place field size remains normal for the hippocampal neurons. So how does C1q actually target the neurons and bring the microglial cells to the synapses? Well, when neurons are stressed and when they the NMDA receptor is engaged, they secrete HMGB1, which is normally a nuclear protein, but can be secreted by activated or stressed cells and then functions as a DAMP. And what you see on the upper right is that the HMGB1 is present in the cytosol of the neurons that were exposed to lupus antibody. And actually, in neuronal culture, you can see that HMGB1 will bind to the NMDA receptor, which had previously been shown by others as well. What you see here is that C1q will not bind to neurons by itself, but will bind in yellow when HMGB1 is also present and binding to the neuronal synapses. So what happens is that stressed neurons secrete HMGB1, which binds the NMDA receptor. And we had previously shown that C1q can bind HMGB1. So the HMGB1 functions as a bridge between the NMDA receptor and scavenger receptors and the C1q, which then attracts scavenger receptors on the microglial cells. So we were interested in whether we could actually dampen the microglial cell activation. So it was known that ACE inhibitors could in fact dampen macrophage activation. What ACE inhibitors do is prevent ACE from mediating the conversion of angiotensin 1 to angiotensin 2, and they also stop ACE from breaking down bradykinin into non-functional bradykinin peptides. So macrophages and microglial cells alike have receptors for angiotensin 2 that can activate the microglial cell. ACE inhibitors are popular and safe in the treatment of hypertension. They've shown potential benefits in retarding the cognitive decline in Alzheimer's disease. It's known that they will decrease interferon production by macrophages. And it's also known that there's a lupus risk allele of the ACE gene, and that this risk allele leads to high ACE expression. So we decided to treat mice with either Captopril, which is an ACE inhibitor that can cross the blood-brain barrier, or enalapril, which is an ACE inhibitor that will not cross the blood-brain barrier. And what you see here is an image of ACE in the brain, and you can see that in the brains where the lupus antibody penetrated, there is much more ACE present than in the brains where normal immunoglobulin penetrated the brain. And so what we did was both a prevention strategy and a treatment strategy. So we immunized mice with either peptide or just with polylysine backbone, so that some mice would be making the lupus antibody and some mice not. And then we gave them LPS. So we would open the blood-brain barrier in the hippocampus and let the antibody through into the brain parenchyma. And we treated either during the acute stage, or we waited until after 4 weeks, until 5 weeks after LPS, when there's already microglial activation and the beginning of dendritic pruning. And we asked whether we could now have a treatment strategy. And what you can see is in mice that were given Captopril, as opposed to mice given saline or enalapril, the microglia have a lacy phenotype and not the compact, stubby phenotype. And that's quantitated in their microglial score below. And what you see is that in mice that were given Captopril, there is now normal neuronal arborization, whereas in mice given saline or enalapril, there is still a decrease in the dendritic arborization in those mice that had lupus antibody penetrate compared to the mice that had normal immunoglobulin penetrate. And this was true both in the prevention model and in the treatment model. So how does this work? Well, our hypothesis is that ACE inhibitors lead to the upregulation of LAIR1. LAIR1 is a known C1q receptor. In fact, we showed that it's a receptor for C1q and that it's an inhibitory receptor for C1q on monocytes. So our hypothesis is that there has to be a balance between scavenger receptors and LAIR1. And when they are well balanced, the microglial cell is quiescent. When the microglial cell is activated, the amount of LAIR1 decreases. There are more scavenger receptors, so C1q preferentially binds to scavenger receptors and pruning occurs. When ACE inhibitors are given, or a ACE inhibitor that will be centrally acting and move into the brain, the LAIR1 expression is increased, and then the C1q will preferentially bind to LAIR1, which exerts an inhibitory effect on microglial cells. And one of the predictions of this model is that Captopril will not help restore normal cognition in LAIR1 knockout mice, because there will be no way to increase LAIR1 expression and turn C1q into a suppressive signal. So when we take microglial cells out of the brain of mice that had either lupus antibody penetrate the brain or normal immunoglobulin penetrate the brain, you see there's less layer 1 expression on those microglia from the brain that had lupus antibody penetrate. And if we just take normal microglial cells and we treat them with HMGB1, which we've shown is being secreted by stressed neurons in the hippocampus of the mice that had lupus antibody penetrate, what you can see is HMGB1 causes a decrease in layer 1 expression. And if we treat wild type mice or layer 1 knockout mice with Captopril, what you see is that in the wild type mice, you see a decrease in microglial activation that you don't see in the layer 1 knockout mice. And if you look at dendritic arborization, Captopril leads to an increase in dendritic arborization in wild type mice, but there is no effect of Captopril in the layer 1 knockout mice. So we think that this model that we've suggested is in fact correct. What we think is that this initial insult causes the surviving neurons to secrete HMGB1, that the HMGB1 binds to RAGE on microglial cells, that activates the microglial cells, leads to a decrease in layer 1 and an increase in production of C1q. The C1q binds to HMGB1 that is bound to NMDA receptors on the neuronal dendrites. That causes dendritic pruning because the C1q is now functioning as an eat me signal. And as the neuron is pruned, it continues to be stressed, continues to make HMGB1. So one has a new pathologic homeostasis in the brain that no longer depends on antibody being present. So how does this relate to patients? Well, we think it relates quite well to patients. First of all, if one looks at blood-brain barrier permeability in patients, you can see that indeed in the hippocampus, lupus patients have a more permeable, a more leaky blood-brain barrier, and this is specific to the hippocampus. It's not true in the thalamus or in the orbital frontal cortex. If one takes lupus serum that has these particular antibodies in it and injects the immunoglobulin from the serum into a normal mouse and gives LPS, these antibodies will penetrate into the hippocampus and bind hippocampal neurons. If you take normal serum or lupus serum depleted of these antibodies, you can see that after LPS treatment, there's much less binding of immunoglobulin to the hippocampal neurons. And if one asks whether these mice have normal spatial memory function, by seeing if they can learn to get out of the maze, what you can see is that the mice shown in red here that have normal immunoglobulin penetrate their brain can get out of the maze much more quickly than the mice that had the lupus antibody penetrate their brain. And if one goes to lupus patients and asks if they have an object memory defect or a spatial memory defect, and one will find a spatial memory defect. So object memory is assessed by asking if there was a dog or an apple in a picture that had 4 images in it. And spatial memory is assessed by asking if two of the images were side by side, in this case, the banana and the lion. And what you can see is that when you do the object memory test, lupus patients, whether they have the antibody or not, perform equivalently to healthy controls. But when one does the spatial memory test, lupus patients that don't have the antibody perform like healthy controls, but both perform differently from lupus patients who have the antibody, who have a spatial memory impairment. One can do PET scanning for glucose uptake, and what you can see is increased glucose uptake, increased metabolism in the hippocampus. This is taken to denote stress neurons in the hippocampus, similar to what we saw in the mice with the neurons that were secreting HMGB1. And if one asks whether this metabolic abnormality associates with memory function dysfunction, it does. It also associates, serum antibodies also associate with memory dysfunction. And if you have both this particular antibody and hypermetabolism, you have a much greater likelihood of having memory dysfunction. If you look at the microstructural integrity around the hippocampus, what you can see is that in those who have poor microstructural integrity, they also have poor performance on a spatial task, on the spatial task I described before. And if you look at the presence of antibody or not at high titer in serum, what you find is that those who have more antibody have less microstructural integrity. So this also suggests that the antibody is related to the hippocampal defect and the poor spatial memory. And if one subjects lupus patients to PET imaging with a PET ligand that binds activated glial cells, you can see again that there is increased binding of this ligand in the hippocampus of lupus patients. We also had been studying neuroimaging in lupus patients and had imaged lupus patients some 15 months apart. So they were imaged once and then they were imaged again 15 months later. And what you can see is that those lupus patients that were taking a centrally acting ACE inhibitors actually had a decrease in their brain hypermetabolism throughout, whereas those mice that were not taking a centrally acting ACE inhibitor did not. So we now have a clinical trial that is just opening, where we look at either an ACE inhibitor that crosses the blood-brain barrier or an ACE inhibitor that does not. And we're asking whether that alters the increased glucose metabolism in lupus patients. Our secondary endpoints include cognitive testing and imaging of activated microglial cells. So where does all this leave us? Well, we think that these antibodies that cross-react with DNA and the NMDA receptor actually do cause cognitive impairment in lupus patients. We don't think they're the only soluble mediator to cause cognitive impairment in lupus patients, but we think these antibodies are important. We think that microglial activation is a key component of the cognitive impairment because it leads to dendritic pruning. And we think this may be a very general phenomenon, whatever the reason for the microglial activation. We think ACE inhibitors are going to be useful, but in a couple of years, I'll actually be able to tell you if that's true or not. And importantly, we think anti-brain antibodies that can set up this pathologic homeostasis are probably present in many individuals. And that in fact, much of cognitive impairment that is attributed to occult vascular disease may actually be immune mediated. It's called occult vascular disease because the radiologist can't see the vascular disease and just assumes it must be there. So this work was done by many people. On the far left are those in my lab who worked on it, currently Jeff Wingard and Mark Mizrachi. The next column has others at the Feinstein that worked on it. David Heidelberg, Ann Vo, and Chris Tang did the imaging studies. Pina Sinelli did the blood-brain barrier imaging. Bruce Volpe and Roseanne Berlin helped us with histology. Cindy and Megan conducted the cognitive tests on the lupus patients. And Pato and his son Tomas did the functional studies of place field size. And then Yoshi and Ichi, now back on the faculty in Japan, both spent time in the lab working on this project. Uma Sriram really introduced me to the role of the ACE inhibitors in suppressing macrophage activation, and we learned to score microglial activation from Beth Stevens. So thank you very much for listening to this. And again, I think what's important here is that we can and must better understand neuropsychiatric lupus so we can treat this devastating symptom.